The ApiNATOMY toolkit has undergone substantial revision since its inception. Here, we first overviewed the original requirements informing early ApiNATOMY design. That's our starting point. Then, we track the evolution in the design principles that recently advanced ApiNATOMY's knowledge model. We see that now it coherently maps detailed representations of physiology processes across scales. ApiNATOMY originally set out to visually provide a broader physiological context to molecular processes, a global map for the molecular economy, as it were, marking out the anatomical locations where molecules are manufactured, where absorbed, where jettisoned, and, above all, where they are allowed to meet and interact in fulfillment of their function. In that sense, the first implementation of ApiNATOMY was simple. It focused on the pharmacological ADME use case. In particular, it provided a straightforward compartmental tree map, a tiled flat map that replicates the topology of partonomy and subsumption relationships found in standard reference ontologies of anatomical knowledge. This tree map approach had limitations, however, as we shall see. The next phase of ApiNATOMY development was initially driven by the requirement to manage knowledge about tissue domains. These domains represent a key choke point between long-range advective transport and the short-range diffusive movement of molecules. With time, the notion of tissue domains was refined to a cylindrical parcellation known as a primary functional tissue unit. This PFTU is a cuff wrapped around a central canal, such that no 2 points within this domain are beyond average diffusion distance for small molecules. This parcellation strategy helps demarcate clearly the 2 key ultrastructures that are relevant to tissue phenotype by separating the sole source of external or endocrine influence, that is the central canal, from the parenchymal cuff, a producer of local signaling factors in its own right. In that sense, the PFTU defines the notion of local in the context of whole-body systems biology. But conceptually, the PFTU did much more than that. As a cylindrical choke point to flow, the PFTU served as a starting example to generalize on choke points across the body. The result? A cylindrical template known as a LIF, to describe conduits that regulate flow at any scale. ApiNATOMY is now being developed to represent the body as a complex of such LIFs, which would include, for instance, large vessel conduits, PFTUs, cells, organelles, and membrane channels, which convey routes of communication that are represented as linked process edges. Representing biology processes as edges is a well-established practice in molecular biology, of course. But the original ApiNATOMY tree map approach needed considerable revision to viably extend this solution from molecular biology to multiscale physiology. For one, visualizing physiology processes as edges requires a graphical as well as semantic relationship with the LIFs that convey them. But the use of a flat tree map that describes conduits also forces edges to pass over structure tiles that are not involved in those processes, creating considerable ambiguity as a result. The generic LIF concept also served to highlight the use of rotation to summarize as a flat map the luminal and mural content for any cylindrical conduit. Early attempts to experiment with rotation on the whole body tree map again had resulted in ambiguity, falsely suggesting that structures such as the lung and the brain are concentric around the gut. The body tree map was not a LIF. So, the tree map as it stood had to go, to be replaced by a more granular assembly of conduit LIFs. This was a radical change in the design of ApiNATOMY, but one that greatly advanced its descriptive ability. So what kind of knowledge can LIF templates capture? Well, LIFs represent conduit structures. In ApiNATOMY, knowledge describing such structures includes annotations to reference ontologies declaring a LIF object as representing an instance of a biological structure class, as well as 4 kinds of mereotopological information, namely 1. Borders and their polarity, given that for any LIF, two longitudinal borders are distinguished by dint of their proximity to the axis of rotation, and two radial borders are arbitrarily labeled for distinction. Borders per se can be annotated with distributions of one-dimensional measurements to capture length and thickness. 2. Composition in terms of other LIFs as parts. So we recognize regions as LIFs contained in another LIF, which may extend to layers, regions that run longitudinally from one radial border to another, and materials, which are aggregates of LIF admixtures constituting the fabric of a LIF. 3. Configuration. Conduits for flow take one of three topological configurations: the bag, which indicates a terminus for flow of material in its innermost layer; the tube, which indicates continuation of flow of material in its innermost layer; and the cyst, a complete envelopment of material flow within its innermost layer. 4. And finally, border sharing, which results from linked assemblies with other LIFs. There are two kinds of assembly: the axial assembly, along the axis of rotation sharing radial borders, and the coalescent assembly, orthogonal to the axis of rotation, sharing longitudinal borders. Let's take a closer look at each type. Axial assembly allows for LIFs to create compounds of arbitrary unit length by following a simple set of operations. Two tubes make a tube, a bag and a tube make a bag, two bags a cyst. These assemblies permit the construction of one of the most important structures in physiology, the arborization, a conduit system such as the pulmonary tree or the blood vessel network that converts flow that is high pressure, high velocity, low exchange to flow that is low pressure, low velocity, high exchange, and vice versa. Arborization assemblies are also imbued with branching probabilities which, in combination with length and thickness distributions, make for a parametrizable model for interspecies or phenotypic differences. These parameters are key for an ApiNATOMY model to emit an anatomically realistic instance of spatial geometry for a particular tree. Conversely, coalescent assemblies allow for conduits to link sideways by sharing the outermost layer of their wall. This approach is well suited, for example, to track compartments for two or more PFTUs, responsible for the regulation of exchange between different arborization systems. So, now that we have the necessary to build a semantically rich, multi-scale scaffold of conduits, let's see some examples of each type of assembly. A first example sees the classic whole body layout as an outer circle of epithelial assemblies for key body surfaces such as gut, lung, genitourinary, and skin lifts, surrounding an inner circle for endothelial assemblies for blood and lymph conduits. And now see examples of arborizations in blue, the urinary arborization, now on the left, and in red, the endothelial arborization for the cardiovascular system. At the very center of these circles is a linear ependymal assembly for cerebrospinal fluid conveyed by brain and spinal cord lifts. This lift-based view of the whole body entirely supplants the original 24-tile tree map in the older implementation of ApiNATOMY. A second example sees a coalescence between an epithelial tube in the urinary arborization and a capillary blood vessel. This coalescence houses the main compartmental layering between blood and urine, an axial assembly of two bags to create the luminal and abluminal regions of a urinary epithelial cell cyst, as well as axial tube assemblies for extracellular, transmembrane, and intracellular portions of membrane channels responsible for sodium transport. So, where are we at with ApiNATOMY development? We are now tooling up to support the collaborative authoring of LIF complexes via a dedicated web app that stores assembly models via a JSON schema and provides a manageable shorthand by which a small set of instructions can build complex layouts of lifts and edges. All code is openly available via GitHub, and the project is actively maintained, extensively documented, and tested to manage knowledge in large multi-scale mapping and modeling efforts. In summary, the advances to the underlying ApiNATOMY knowledge representation we've discussed now provide the means to extend classical molecular biology network depiction to represent process knowledge in multiscale physiology by explicitly relating a transfer process with the conduit that brokers it, thus latching ApiNATOMY's visual metaphor to a representation prevalent in molecular biology, that of an enzyme catalyzing a reaction. Also by allowing for typed edges to represent different kinds of process. In that sense, we now have the option of either leveraging large numbers of qualitative descriptors from a reference ontology of biological processes, such as the gene ontology, or importing a pre-existing formalism for the quantitative description of process graphs, as found, for instance, in the bond graph formalism. And also by assembling complex conduits to achieve a key representational milestone as yet unattainable in classic molecular biology schematics, namely, the dual ability for a biological object to feasibly have both a participant role in a process as well as the role of compartmental location for other processes within it. And with that, we continue on our path to develop more robust and useful tools in physiology knowledge management, thanks in no small measure to the support of the neuroscience and physiology communities. We urge you to follow our progress via our documentation site, as well as our GitHub updates. Thank you.